Hello and welcome to a webinar from inviCRO on some new imaging techniques that we've been working on. My name is Jack Hoppin. I am one of the co-developers of this effort, a team of many who we'll thank later. And the topic itself is on high-throughput three-dimensional tissue imaging in the bright field, fluorescent, and nuclear regimes. We're going to explain what that means. We've had some pressure for quite some time to talk about this. We've presented it at conferences, and we presented it at we've presented at conferences, and we've presented it a few places. And just as a point of update, we wanted to give a webinar so that people knew what we were doing. You know, if webinars are not that fun to give because you're just sort of talking at your computer, so bear with me, and thank you for listening. But we felt like if we didn't explain what we were working on, that of course no one would actually know what we were doing. So as a point of, hold on, going to the next slide, just an overview, just give a quick update and outlook on inviCRO. Try to motivate where, why we started working on this effort, then go through some of our early work, which was really around the bright field and nuclear, 3D nuclear efforts, much of which was inspired by some work that had been done a long time ago at Wyeth. Fluorescence is sort of the main takeaway and effort of this talk, where we're starting to do what we're calling cryo-fluorescent tomography, or fluorescent imaging off of a block surface. By block, I mean a frozen block of tissue, whether it's for just a resected organ or a larger whole mouse or whole rat. And we'll sort of highlight the upside of this in general is about multi-resolution imaging. And we'll talk about the outlook of the technology. So briefly, as an overview of the company, so inviCRO was founded in 2008. We're really an imaging informatics company. We had a background in the founders had a background in imaging system development in the nuclear, in the SPECT CT space. We started the company out the gate. We had two softwares that we provided, IPAX and VivoQuant. VivoQuant has had many names, the MIP tool, but today known as VivoQuant. And we got big enough. We started in office space, and ultimately grew to a size we were able to open up our own imaging facility, which is own preclinical imaging facility, which is based here in Boston, where I'm sitting now. We've effectively a rodent radiology clinic here, and we also opened our European headquarters in London in 2013. We jointly opened a translational imaging center at MPR Research for mouse to non-human primate imaging, and after many years of doing research with the imaging group at MPR Research, a great imaging team there, we founded that in 2014. That's a cyclotron-enabled facility. We installed the cyclotron there at that time. And sort of in biggest news in February of this year, molecular neuroimaging joined inviCRO and became our clinical division. I'll talk about their facility on the next slide, but now all in, we have really a very strong and vast team of imaging researchers. We have over 200 employees, most of whom are working on a daily basis as an imaging researcher or supporting an imaging trial. We have 25,000 square feet in Boston with this imaging facility. We have this 20,000 square foot facility on the campus of MPR Research that we jointly support and manage. We have 30,000, the M&I facility, the clinical division facility is 30,000 square feet, a great team in New Haven. We've run a lot of preclinical and clinical imaging trials. The team in New Haven has submitted more than 60 INDs for first-in-human imaging. We process a lot of data. We have, and not shown here, but the New Haven team has over 700 imaging sites under management for clinical trials, and we're happily working on some of the most exciting imaging research projects in the drug discovery and development place. So this is just to reiterate sort of the overview of those facilities and capabilities. And again, underlying all of this is a drive to be a quantitative imaging company for those doing drug discovery and development. If we look by division, we have a preclinical space here, this first in human, very unique clinic in New Haven. The clinical imaging core lab is in New Haven, and part of the marriage of these two groups was driven by marrying the software and capabilities in Boston with the clinical core lab capabilities in New Haven. We're having a fun time doing that. A lot of research, a lot of radiochemistry, huge radiochemistry group in both research and CGMP chemistry group in New Haven, and research chemistry groups in Boston and in Michigan, and as well as a network of many other teams we work with, including the London team, and a core competency in image processing. We process a lot of image data here. And it's the image analysis and software teams that really are the drivers of this new technique that we're presenting today, where we think we have a useful, we'll say is supracellular sub-organ microscopy strategy. I'll say that a little bit again later. Here's our view of the planet, working from a biological question, having a strategy of design, execution, raw data. The fun often is in taking the process data and mapping it to doing a dimension reduction to an array of numbers that we can then run through stats, plotting, and reporting tools. And we've dedicated our development efforts to having software packages and a research team here, a diverse research team to really support any aspect of this workflow for a primarily pharmaceutical driven research sponsor. This is a schematic diagram that we recently published with Richard Hargreaves and some of his team from Merck and Biogen. And you're looking at from validation to clinical use, and we really have spent a lot between Boston and New Haven, London and Michigan. We spend most of our time right in that middle area, compound screening, preclinical testing of lead compounds, and then first in human trials, and then supporting phase 3 trials as the clinical imaging core lab. Or some really exciting multi-center, primarily AD and PD trials right now. And our perspective on the sort of imaging trials that come our way is we usually bucket them into one of 3 buckets: biomarker imaging, so either developing an imaging biomarker, reading out working on a quantitative imaging biomarker at any step of the process, as you can see here, developing a new one, or literally putting one to use in a multi-center clinical trial for a readout for a therapeutic. Biodistribution, so putting a radio label, most commonly a radio label, or as we'll show you today, a fluorophore, some signaling moiety onto the therapeutic itself, beta, gamma, alpha even, but mostly beta, gammas, or fluorophores, and then trying to image its distribution. And lastly, I bucket into reporter gene strategy, so having some fluorescent protein or some means of visualizing gene expression. And we haven't done a lot of that historically, although it's very, very popular, as everyone knows, in the preclinical space. But I think we have a useful technique now in this 3-dimensional fluorescence capability that we'll present today that kind of expands potentially the role of reporter gene strategy imaging in the marketplace and in the research community. Okay, so this is a plot also from the same paper looking at resolution, volumetric resolution versus mass. And I'm going to stop here because we actually had audio-visual challenges, and this is a repeat of the first 7 slides. I just repeated them. I'm going to stop, and you're going to pick up on the first broadcast right after I stop. You're going to hear me again in 1 second. Okay? I hope you enjoy the rest of the talk. Oh, I see. Is quantitative whole body, or excuse me, micro autoradiography. We had a technical difficulty here. We've overcome it. Right in the middle, you see we've circled in vivo rodent nuclear tomography, which is really only about a 15-year-old activity and served as in a high level. I'm focused on nuclear imaging on this slide, but really if we look to the left, that's pathology, and to the right, that's radiology, and there was quite a gap between, and still is, those are pretty siloed departments clinically, but between imaging, histopathology, imaging ex vivo tissue imaging, and in vivo medical imaging that you'd find in the clinic. And one thing that this in vivo rodent tomography enabled was the ability to, in some ways, expedite the translation of techniques from the bench to the clinic. Go to the next slide. Okay. And part of the fun of our job is looking at from a molecular imaging perspective, the huge array of sensitivities that are afforded to someone to do molecular imaging. If we look at the red, I won't belabor this plot, but if we look at the red boxes, we have micro autoradiography, PET, and SPECT. This is just a revamp, but now looking from the sensitivity perspective, that we can go from a cell to a human and at very, very high sensitivity, which is why we love nuclear imaging. We would love to do more fluorescence imaging, but it's limited in the scope of which one can interrogate, just simply because of the absorption of the photons in tissue. And I'm going to sort of speak about today somewhere in this 100 micron, trying to expand our ability to do 3D fluorescence and 3D nuclear and 3D bright field imaging. I alluded to earlier that we were personally active in the early days of doing three-dimensional gamma ray tomography, SPECT specifically, but very active in PET and as well. And if we look over the last 15 years across the whole electromagnetic spectrum, all of these, the whole gamut now is available in rodents. Tissue imaging pathology is 100 years old or longer. Radiology is 105 years old, according to the RSNA this year. And what's happened in the last decade or so is we've had the computational power and the detector capabilities to reconstruct images in rodents across this spectrum. In the top, you see radio wave, MR imaging, ultrasound, and down the bottom, gamma ray and CT. But in the middle, we haven't done a lot of whole rodent bright field and infrared imaging, and that's what I'm going to sort of highlight as new capabilities that we offer here today. Most of our work, what we like to do most often is radiolabel a drug of interest, administer it to an animal, image it dynamically, in vivo, thus inviCRO, in vivo, contract research organization. We like to image it dynamically, and then we often take the animal down and do autoradiography. This takes us from a microliter or cubic millimeter sport in vivo to a nanoliter type of resolution in autoradiography. So we're 1,000 times better ex vivo, but we're, of course, the tissue is the animal sacrificed. And it's sort of something we'd like to do in fluorescence as well, and that's what we'll go to. But before we did that, we started, and this was work initially inspired by Stefan Linehan, who started this work many, many years ago at a pharmaceutical company, and that was taking bright field images off the block at each depth and reconstructing in 3D the distribution. So there you see the bright field image of an animal off the block, and we're actually able to reconstruct in 3 dimensions the red, green, blue images at each depth. And while this is clicking automatically somehow, while
while I show their sagittal views, we actually can reconstruct in 3 dimensions. So we're capable here of looking at a transverse section, as you see here, in the base of the spine, and you're actually able to see. For some reason, it's just like auto clicking. You're actually able to see a cross section of the. In this case, we'd administered Evans blue. You're able to see the nerve fibrils running through the base of the skeleton. We've done this across a lot of species. It's provided a lot of very useful anatomical information. Here you see a whole cynomolgus head that we reconstructed. And I really show this for 2 reasons. One, it is a striking image. It actually was very useful in discerning some nasal anatomy in this case. We did actually administer a radiolabeled compound here. I'm not able to show you that. But I do want to state that sort of as a breakthrough component, what's happened here is you have an 1800 by 1800 by 1800 3-channel RGB reconstructed data set. This takes about 55 gigs of RAM, which is now possible on a desktop. That would not have been possible as recently as, well, certainly when we started inviCRO. A lot of the driver here of what we show you today is inspired by computational power, the ability to store so much data on disk on a regular computer. It just was not possible in times past. This is effectively 40 micron 3-dimensional isotropic resolution through a very, very large field of view. The other advancement here is just having, as we'll show sort of a detail later, is a 3-dimensional RGB viewer, which was a 3-dimensional RGB medical viewer. We'll focus on that as sort of something we did a lot of work on. So first example I'm going to give is a nuclear example. This is now looking at an I-124 and I-125 labeled protein looking dynamically in a dog paw. This dog paw was a Pancarpal Arthrodesis model, and we've applied the radioactive drug in paste form in the dog paw. And on the left, you're looking at the PET CT of the paw, and on the right, you're looking at the dynamic washout of the protein, the labeled protein, over the course of 2 weeks. Because we had put in a I-125, we actually were able to track this study out 5 weeks, and at the end we took some pause, which I would add was very good. First of all, had previously been a 50 hound study, and we were able to just do 2 cohorts of 5 animals, so a huge, and this is a case where longitudinal in vivo imaging really required far fewer subjects. In this image here on the left, you see the bright field image, you see the dehydrated section, and then you see the autoradioluminogram with the I-125 hot sections from a phosphor imager. We do this at multiple depths, and we're actually able to reconstruct in 3 dimensions the 3D RGB view of the dog paw, as well as the pseudo-color rainbow scale, in this case of the iodinated protein. So this was pretty cool. We were very, very excited about this. Again, back from a resolution perspective, the iodine labeled PET work is on the order of, 10 microliter type resolution, and we're looking here on the 10 nanoliter type of resolution. So a huge gain in volumetric resolution, and very quantitative, and gave us a good insight into the distribution in just a couple samples at 5 weeks post-administration of the drug. So very exciting, very useful, but I'll be blunt, very hard, very tedious, and very time-consuming. While there's work being done that we're active in trying to image radio image betas and alphas directly off the block, as it stands now, you still have to transfer the tape, put the tape on a phosphor imager, and then expose, read out, reconstruct, and to do 3-dimensional autoradiography can be very useful, but can be very time-consuming. The anatomical imaging, any way you look at it, here you see the a whole view of the dog paw is striking, very useful, and this can be sped up, and I'll show you later how we're able to collect the 3D optical images in much faster time frame. Just another application, a dual isotope application, which I think was very, very useful and very useful to anyone doing ADC work. This is antibody drug conjugate where we indium labeled the antibody, and the drug conjugate in this case, the warhead was tritium, was synthesized with tritium. So what we're looking at here is 96 hours post-administration of the ADC. There's an antigen positive tumor along the top, an antigen negative tumor along the bottom at the same time point. And what you're looking at here is the tritium, deposited tritium through one slice. On the left hand, that's the three-dimensional reconstruction using this technique of the xenograft, resected xenograft. We took autoradioluminograms at many depths, of both the indium as well as tritium plates. We then superimposed them here in the middle, and you can see that the overlap at that time point is not exactly what you might expect. They're not fully superimposed. So just not diving into the depth of the trial, but it was interesting, and we were actually able to quantify that difference. You can see here in the positive and negative tumors, there's great separation between the tritium and the indium in the positive tumor. Whether you hypothesize that just the antibodies deposited the warhead and moved on or whatever, that's beyond the scope of this meeting, but just very useful, and for one of the first times at a very high resolution in 3 dimensions to be able to distinguish where the antibody and the actual deposited tritium warhead were at that time point. And very differently, you see in the negative case, there's a lot of overlap, and that's really largely just vasculature. Okay, so now to the main event. Although we really do like 3D, and when we say block face here, we're really talking about taking resected tissue or whole animals. Resected tissue or whole animal blocked and cut on either a cryostat or a cryomacrotome. You could also do the work I'm showing you on a microtome. We just don't show any of that work today. This is all fresh frozen work, and we subsequently call it cryofluorescent tomography in-house often, but it doesn't have to be cryo. And we, again, back to this supracellular suborgan, I'm going to show you both supracellular and subcellular images, but the process is really about doing higher throughput supracellular imaging. The last plot from that review article that sort of outlined the scope and breadth of these imaging technologies was one we focus on fluorescence imaging. And here you see, again, volumetric resolution versus massive tissue interrogated. And there's 2 clusters. The bottom left is all of the fluorescent microscopy techniques, and the upper right are the in vivo fluorescence techniques. And there's again a gap between the 2 technologies, and this is really what we wanted to address. Here is, for those, I'm sure many of you are very familiar with in vivo fluorescence imaging, much more common than in vivo nuclear imaging. To the 500 rodent nuclear scanners in use around the world, there's probably 2,500 to 3,000 or 3,500 fluorescent scanners used for primarily bioluminescence, luciferin luciferase imaging, as you see an example on the left. But people also very commonly will put a fluorophore on a drug and image it dynamically. You see an example of that on the right. Or they have some reporter gene, GFP most commonly, where they're going to try to look at the gene expression and visualize it in vivo. And that's the type of surface images you get. There's a lot of work, and we've spent a lot of time on it ourselves, trying to do 3-dimensional reconstructions from the images, whether they be bioluminescence or fluorescence. And while a lot of work's been put in, I can't say we as a field feel as if we achieved great 3D bioluminescence or fluorescence reconstructions in vivo. We had a system from the Frangioni group called the K-Flare, still have it here, And we were doing some surgical applications with this. It's a fluorescent guided surgery instrument. Here you see indocyanine green injected IV, and we're just collecting a movie of a mouse IV. You know, interesting, you see it clears renally. You can't quantitate anything here. You could maybe try to draw some ROIs around the kidneys and try to get some time clearance renally, but by and large, it's in a non-invasive environment, it's quite hard to get a big read from surface fluorescent techniques. Well, we were already doing block-based imaging, and what we did was we hacked the Curadel Franchione system, and we built a stand, and we mounted it adjacent to a cryomacrotome. So what you're seeing there on the screen, for those of you unfamiliar, is a cryomacrotome. I've talked about cryostats, and there'll be a picture of one later. I think many people are familiar with cryostats. They're almost ubiquitous in the research environment, whereas the cryomacrotome is about literally 100 times less prevalent. You can see a block there next to that paintbrush. That's the frozen block of tissue. It's about 10 by 15 by quite long. It can be quite long in width. That's variable centimeters in dimension. And you can actually just see, if you look in there, I think those are mouse torsos vertically placed, and you can, the blade comes through sections. And what we do most commonly, if you see in the upper right-hand corner, there's some pink tape. The most common thing we would do for quantitative whole body autoradiography is put the tape on top of the block, slice through the block, take the tape off, put it on a frame, dehydrate the section, transfer it to a phosphorimager, and get an autoradioluminogram. This is a technique that's been done for quite some time and is very standard in safety tox, gen tox, and discovery work, primarily with carbon-14 and tritium, though we, of course, can do many other radionuclides. Well, what we decided to do, as you see here, is hack the system. We've 3D printed a holder there, and we started taking fluorescent images off the block. And the first thing we ever did was fluorescent string soaked in a fluorophore. You can see it in an OCT block. This is actually done on the cryostat, so a much smaller block. And you see in the black and white image in the upper corner an image of the string, and you actually get excitation. The laser penetrates the block, so you actually get signal from below the depth, below the surface, which quote-unquote ruins your image. However, since we're sectioning through the block, we're actually able to account and reconstruct in 3D the final string. This is what we've submitted a patent on, and that's sort of the challenge. You get blurring at depths, and you have to collect at each depth an image of the bright field and in this case, near infrared, and then you're able to reconstruct in 3 dimensions. You see a very gloriously reconstructed individual twine strand on the right with our reconstruction algorithm. So if you do whole animals without reconstruction, you'll see there's sort of a general blurring through the animal, and then you apply this 3-dimensional reconstruction. And the first image we ever did was a fluorophore labeled compound that we administered intrathecally. You can see it here. This is a rat brain. It was a proof of concept, and to be honest, it was just a lot better than we had expected. We figured we'd just try this. Let's see how it goes. And if you notice, also on this image, this brain is perfectly symmetric because we actually only did 1/2 of the brain and then digitally flipped it over to the other side. But nonetheless, we were pretty excited. You can see a very high resolution reconstruction of the fluorophore. Well, we worked on this further. Again, you see on the left, we're not only getting the reconstructed fluorophore, but also the reconstructed anatomy of the bright field image of the rat brain, and we've co-registered those. And that was a non-trivial endeavor, both on the reconstruction side as well as on the expanding the VivoQuant to support the RGB viewer, and then co-registered with the pseudo color scaled fluorophore in this case. For those of you who are less familiar with this concept, you can think that all medical imaging in general is a DICOM pseudo color sport, 16-bit sport where you map to the full 16-bit coming off the camera, and usually with some scaling factor. Here we actually are presenting all 3 channels and in one space, and then the pseudo color in the other. So it's quite nice. You have actual anatomical images in one image superimposed with the fluorescence, and it's in the medical viewer. So you can go register with other items, you can quantify, you can segment, you can and do everything a regular medical viewer can do, as compared to a classic. Not all Z-stack microscopy, confocal microscopy would have full medical viewing capability. Okay, so I'm going to show you here one of the first studies we did where we were struggling with getting a radionuclide onto a therapeutic, but it had been synthesized with a fluorophore. And so here we look at the result of 6 animals administered, 6 rats had been administered with this therapeutic compound intrathecally, and we were able to reconstruct the fluorescence in 3-space using this technique. Very striking resolution. No one had seen the distribution of this fluorophore at that resolution. Again, this is something on the order of 25 micron voxels. So we were not accomplishing subcellular resolution, but we are accomplishing very, very high resolution suborgan resolution. And you can sort of map out through a series of ex vivo scans, you can get a time course. And again, you get a 3D view of the anatomy as well. But very exciting. I mean, you can really kind of see the drug enter and start to wash out of the brain. What's really nice here as well is you can, at any given time point, as you're sectioning, again, if you look across the top, this cryofluorescent tomography, we were sectioning in the, it depends on how you want to call it, but we'll say the transverse, or everyone likes to debate what's corona and transverse in a rat brain, but whatever you want to call it, the sectioning on the right, but you reconstruct in all 3 dimensions, of course, but then we can actually take tissue. In this case, we just took it to glass slide, and we can put it under a fluorescence microscope, and we're co-staining here for DAPI and compound. Taking it a step further, we can show it going from the super organ down all the way really to a zoom in, showing that the compound is co-localized with the neuronal nuclei. So really across a huge array, a volumetric array. And I'm going to talk about speed a little bit later, but the idea is it's quite, to be honest, quite quick and easy to do the whole brain. And it's actually, of course, as you know, immunohistochemistry and histopathology is more time consuming, but very useful, right? And we can co-localize them here. So that's really everything I've shown so far has been on the cryostat with resected OCT blocks. And I just want to remind you, we have here at our ready these cryomacrotomes. So we're able to do a 3-dimensional reconstruction off this block, just as we did in the nuclear. An example I'm going to show you, which many of you I'm sure are familiar with this biology, more familiar with me. I also want to state I'm neither a biologist nor I'm new to the field of histopathology in general, so if I butcher anything on that front, please forgive me. I come to you as an imaging scientist in this context, but again, I really I'm very familiar with these applications and really think that this technique is useful, or I would not be presenting it to you. You see here just protoporphyrin 9 production schematic to make heme, the most common source of background really in most of our fluorescent imaging capabilities. But if we give 5-ALA, this is used for photodynamic therapy, and what we're going to show here, because that'll really increase protoporphyrin 9 production, what we show you on this slide here is a whole body image of so this is a mouse in a block, and we're looking at the whole body. This is the protoporphyrin 9 production just inherently. You have the Harderian glands, you have the GI, you can see actually feces down at the bottom of the screen in the animal. And now if we administer saline, nothing changes, of course. If we administer ALA5 as a precursor, we're going to see huge ramp-ups in porphyrin production. So there you see a whole spinning MIP of the ALA5 production, and we're imaging this off the block in 3D. So from my experience, given the speed and scope of this, to do 3D fluorescence like this is so quickly and cheaply is really useful. Again, we can then go to fluorescent microscopy after the fact, but here you're really seeing the whole distribution in the animal. And while we only show the distribution of the fluorophore because it's easiest in a MIP rendering, and inviCRO loves being made fun of by how often we show spinning mice, and we felt obligated to show a spinning mouse, you do, of course, have the co-registration fly-through of the RGB with the fluorophore distribution. So we were pretty excited about that. And you can, as I alluded to, and I just wanted to show you that it really, you can load and visualize these items in 3D. So it can be really, really useful to draw regions of interest, to co-register with other items, to view in multi-spaces. And so that, for sure, has been a great, we've put a lot of work into that. All right, but the problem still is to date has been that it's tedious. So realistically, it's been, and you can imagine how tedious it is to do 1,800 digital photographs off a block and then reconstruct them, taking sections via tape. It's all been very hard, and please, and a huge shout out and thank you to everyone who's worked on this effort, and especially those who spent a lot of time in the lab. Ildiko Polyak stands for, I don't want to cut off any names, but many people have spent a lot of their, Paige Charnetsky, a lot of people spent a lot of their time working on in the lab on the system. So we were, we of course brought this finding back to the Frangioni group, to Curadel, and said, hey, how can we work together with the software we've created along with the hardware you have, and build a system that you can modify your fluorescent guided surgery system for better use in this application. And here you see the first schematic of the fluorescent guided surgery system on the left and the cryostat on the right. That got even fancier when a color schematic was made of the same thing. And then as a realization, here you see, and I'd say the only thing missing from that previous schematic is this nitrogen air gun that we add in order to blow tissue off the block as we're sectioning automatically. You see there's a motor here, a stepper motor that slices automatically through the block. Here's the camera. You have currently the bright field as well as 2 NIR channels. It's also being outfitted for blue and green currently for the next gen that we should have in our hands within around 6 weeks, which we're very excited about. It's a much higher resolution system than the previous gen. The K-Flare was, this is 1024 by 1024 with zoom into basically a square centimeter out to 13 by 13 centimeters. So really, really flexible capability. Really exciting. But I'd say probably the biggest, part of the biggest game changer here is just the automated sectioning. So we can do around 500 sections in 30 minutes. That is really convenient. And I'm going to show you some of the first results. But again, the order of operations would be section, or take a digital, take a scan, slice, air gun away any debris, repeat, and collecting automatically through. Now on the cryostat, we had to build the stepper motor. On a cryomacrotome, we've actually hacked that system, and a cryomacrotome is actually easier. You can override the system and put it in an auto slice mode, so you just have to run the controller to do the air gun and the digital acquisition. We've done that, but not yet with this newer, higher resolution system. So those previous ALA scans, ALA5, the porphyrin scans I showed were very good, but they're probably 10 times worse volumetrically than what we would be able to achieve with this system. Here you see it sort of as just an anatomical view. Just to be clear, that's the older system on top. Sorry, that's the older system in the top left, bright field, as well as the fluorescence with the new system, just much higher resolution. I want to be clear, some of the anatomy, some of the anatomical images you saw earlier, we actually had mounted a separate digital camera to the block, not just the system from the fluorescent guided surgery instrument. So you may, for those keeping score at home, you may have noted that some of the anatomical images were higher quality, and that's just caused by that. Lastly, last but not least, this is this is exactly the goal of what we were trying to accomplish. Here you see two compounds injected with fluorophores, one ZW800-1, one ZW700-1. Their names indicate the relevant wavelengths at which we're collecting. And you administered simultaneously, and we're able to look at, really the proof of concept. We show here compound A at one wavelength and compound B at the other. Very striking differences as to where they are in the renal clearance. You see the very high res, again, super organ, sub-organ supercellular. So these are not subcellular images. They're at this 20-some micron voxels. I'll talk about resolution in a second, but you can really see some nephron. You see the pelvis, you can see it leading to the ureter, and on the right you see the medulla very clearly heading to the pelvis. I mean, from an anatomical perspective, these are very striking, and this image can be created in around 25 minutes. So I think if I just shamelessly put on my researcher hat of why we want this technique, to be able to do whole organs that quickly as compared to multi-week immunohistochemistry or histopathology is awesome for us. So we have a lot of biologics that come through here. We've radio labeled hundreds of biologics, so the ability to actually just put a fluorophore on a biologic as well, or exclusively a fluorophore on a biologic, is really useful. So I think that's been a fun, we're just pretty excited about the volumetric resolution you're seeing there. I want to be clear, people love to state, and it's a pet peeve of mine, people love to state the voxel size as the resolution of an imaging system. Most commonly in micro CT, they have 20 micron voxels, they claim 20 micron resolution. That makes no sense at all. We would not claim to have 20 micron resolution here with this configuration. We hope to trend to that resolution with thinner sections and a zoom in. That should be possible, but in practice, it's a variable zoom on the system, so it's really it's your volumetric resolution. Your imaging resolution will go as the volume that you interrogate. But I always like to, for just rule of thumb, state, I think it's prudent to state if we have 20 micron voxels, we probably have something like 80 micron to 100 micron resolution is more realistic. And then here is just a resected lymph node. Showing you really showing off the capable. We had something where we had lymph node drainage, and we resected a block encasing the lymph node, and we did a fly-through image of a lymph node, and we can see the drug moving down into the lymph node. So very, very striking capability. Again, very cheap and very fast. So in terms of outlook, I just wanted to talk about where we stand on this project. And again, the sentiment for this webinar was we've had a lot of talks with a lot of people, and people keep asking us to sort of summarize where we stand. If you weren't one of those people and you came out of left field to this webinar, I apologize. I hope I covered what we've been doing. But if we don't actually tell people what we're doing, they can't guess. So the sentiment of the webinar was just really to state where we stand, and where we're ready to engage on this technique. We're looking, again, of course, we're trying to support the commercialization of this technique. We have the first installations of the turnkey system going in this summer and fall. We're working hard to have a 3D quantification capability on the resolution. Right now, it's a relative scale based on a fiducial. We've added, in addition to the bright field and the two NIR channels, we're adding blue and green. I cannot show you, though we have done three-dimensional IRFP, I just do not have the ability to show you gene, in this case, a vector that was deposited, like literally imaging gene expression with IRFP. We have done that in 3 dimensions. I just don't have permission to show it, but that is really, we think, in addition to the straight up biodistribution. That's a huge application here. The other thing we have done, also I'm not showing it here, is we've just administered antibodies systemically that are used in immunohistochemistry to see if we could then visualize them in 3D as a form of doing 3D immunohistochemistry. That has worked in the 2 cases we've tried, but early days. And part of the reason we're giving this webinar is, of course, if you have any other applications you can think of. I admit personally I'm really from the nuclear space and the nuclear, really from the tomography space. And so it's been exciting to work on this and learn about fluorescent, both gene expression as well as straight fluorophore applications. I think that we have the speed down, or we're really a long way to the speed for the cryostat. We look forward to doing the high throughput whole block, whole animal imaging off the cryomacrotome. As you saw there with the saline, one thing we really like is that you can actually put a control animal in the block, so you know what is autofluorescence as compared to what's actually the administered fluorophore. So that's an exciting use of the block. And then on the multi-resolution front, I have one more thing that we've done a lot of work on that I think is very exciting, we showed you earlier taking a section, putting it to a glass slide, and doing my fluorescent microscopy with it. The morphology in that instance is totally destroyed. It's very hard to transfer tissue to a slide and co-register it with any tomography, whether it's this new type of tomography or MRI, or it doesn't really matter. It's hard for multiple reasons. One, the resolutions are very different. That's one thing that can make it challenging, but more relevantly, the tissue is just inherently going to move. Here we have a technique where we've co-registered tissue from the tape. By transferring the tape, you really have a much better chance. And this is early days, and we were very excited about this. And I'm going to show you why it pales in comparison to what we have now. But here you see this is an inorganic packing tape, and we've stained with hematoxylin the isin, the E. I don't even know the right pronunciation of that stain. Does not work because it just doesn't work on this tape. If you look at the pseudo color scale of the you look at the hematoxylin image here, you see a lot of the tissue will inherently dehydrate on the site. That's what you do. You dehydrate the tissue, and that also causes like a local morphological change that can ruin the co-registration. The general co-registration here is very nice, but you on a local level you really struggle because you've had this dehydration. We found, and I would love to meet this person someday. They're in Japan. Kawamoto, the Kawamoto lab in Japan has worked for a long time on a type of tape that allows you to transfer to this Kawamoto tape. It's on the next slide, and it's very striking. So we're able to transfer the tissue to the Kawamoto tape. It's an organic tape. We can actually stain, in this case H&E. We've also done immunohistochemistry on this tape, and you can see that it's preserved very well here, the tissue, the anatomy, which is a huge win for us because we can do this, our 3D block-based imaging, and then as we see fit, section to tape and do staining. Here you can see this is off tape, but when you zoom in, it really is very nice H&E. I mean, it won't be as nice necessarily as a paraffin, embedded glass slide strategy, but it's still pretty great. And so that's something we're excited about now, that you have a full automated fly-through. You can currently, the first system will be to stop, transfer the tape, do the histopathology, but it will be co-registered in the context. You just record what section you stopped at. We'll then co-register that within the context as of the whole 3D RECO, and then as you fly through in the viewer, you can then stop, load that, and zoom in and zoom out. That's what we're working on now. I think we'll be done with that for sure this year. So that was my 47 minutes, and here I really want to thank a huge group of people that worked very hard. It's been a huge team of people here. at inviCRO, as well as a phenomenal team at Curadel. And I'm sorry if I cut any names short. They're tremendous co-workers. They've been amazing in their engineering and turnaround. And I really also want to thank Biogen, and actually Daniel Bradley, now Biogen, previously at Takeda, who sponsored the tumor work. I want to thank them for their sponsorship and driving the development of these techniques. So thank you for your time online. Happy to take some questions now and happy to talk anytime. The first question is, asking, can you show up? Nice present. Thank you for your kind words. What about radio-labeled compounds for PET? Do you do custom synthesis, or do you have a ready list? What ready lists do you offer for imaging purposes? We, I did not drive through our any of the details of our service offerings per se, but yes, we have 30 radiochemists employed here, of which about 15 or so are in the lab. We do a lot of PET synthesis. We do a lot of PET autoradiography, and we in this vein, but also ex vivo binding assays. This is as you saw on like slide 3, this is a chemistry company now all the way. Gilles Tamillon, is the head, but there's a lot of great chemists in the organization, and we do own a cyclotron. So as far as where we can image right now, we can image size 7, and then we have I think the lasers at 670, imaging at 700, 770 to 800. And there's a blue and green going in, and I don't actually have the exact numbers on them. We haven't tested them yet. And the next question is the typical data size. Yeah, it really varies a lot. But tens of gigs to as small as half a gig. Just depends on how the field view interrogated and what you'd like to look at. The question next is, what role do you see this system, new technology, playing in the drug development pipeline, particularly in complement of standard autoradiography techniques? I think that for as compared to autoradiography, I think we've shown through time that the 3-dimensional autoradiography is a special case scenario for autoradiography. It's not needed all the time. Again, it's going to be challenging. If we could automate it, that would be a totally different conversation. But as it goes, it's very labor intensive. When it comes to the fluorescence, now that we've automated it, it's just I think my prediction is that it's going to start as a very a regularly used assay for, again, reporter genes, biomarker strategies, and really most commonly for biodistribution studies are our early requests. Let's just put a fluorophore on this antibody and see its 3-dimensional biodistribution, and 4-dimensional if we take down animals at multiple times. That's a very straightforward application to understand biodistribution. But I mean, it's just very useful. Also, adenovirus, anything that's you're trying to image gene expression, we can now do that in 3D. It's so useful to have the 3D anatomy instead of just a section on a slide. That's my opinion. And I think that is the end of our webinar. Again, I want to thank all of you who attended, and I really want to thank everyone who worked hard. Out of this list, it's such a team effort. There's so much chemistry. There's so much lab work. There's so much study management. There's so much sponsorship. And, of course, there's a lot of engineering on the part of Curadel and a lot of software development and image reconstruction work, algorithm development here at inviCRO. So thanks so much for everyone's time. And my other thing is, you don't need to, just a shameless thing, we are looking for samples. If you have samples, if you're doing any fluorescence work, if you can just reach out to us. Yes, the system will be available. But in the meantime, we're really trying to ramp up different applications. If you have any ideas for applications, if you're doing any fluorophore work, again, the green and blue will be soon. Just ping us, but the near-infrared is up and running. We're looking to expand, we're looking to do work. So please reach out.